Welcome, curious minds, to a journey into the most profound and mysterious realm on our planet, the deep ocean. For centuries, humanity has gazed upon the vast expanse of the sea, imagining what wonders lie beneath its glittering surface. Yet, for all our advancements, the deep ocean remains, in many ways, more alien and less explored than the surface of the moon, or even the distant reaches of Mars. It is a world cloaked in perpetual darkness, under crushing pressures, and swept by currents of immense power. And it is precisely this enigma, this raw, untamed frontier, that forms its irresistible lure. Consider for a moment the sheer scale of this hidden world. The deep ocean, broadly defined as everything below the sunlit surface layer, comprises over 95% of Earth's living space. More than two-thirds of our planet is covered by water, and the average depth of the ocean is around 700 meters, or over 2 miles. If you were to flatten out Earth's landmasses and push them into the deepest trenches, they wouldn't even fill them. This immense volume of water holds secrets that continue to redefine our understanding of life, geology, and the very processes that govern our planet. It is a place where every discovery feels like a revelation, where the boundaries of what we thought possible are constantly being stretched. What kind of world is this, where light from the sun cannot penetrate, where temperatures hover just above freezing, and where the weight of the water above exerts pressures equivalent to dozens of jumbo jets stacked upon a single human thumb? It is a world that demands extraordinary adaptations, pushing the very limits of biological possibility. Far from being a barren wasteland, the deep ocean teems with life forms that are often bizarre, beautiful, and utterly unique, perfectly evolved to thrive in conditions that would instantly annihilate most surface-dwelling organisms. From delicate bioluminescent creatures that craft their own light shows in the darkness, to massive, slow-moving giants and highly specialized organisms that rely on chemical energy rather than sunlight. The biodiversity of the abyss is a testament to nature's boundless inventiveness. This journey into the deep isn't merely an academic exercise. It's an exploration vital to our own existence. The deep ocean plays an indispensable role in regulating Earth's climate and chemistry. It acts as a massive carbon sink, absorbing vast amounts of carbon dioxide from the atmosphere, helping to mitigate the effects of climate change. Its currents distribute heat around the globe, influencing weather patterns from the poles to the equator. Furthermore, the deep sea holds untold potential for scientific breakthroughs, offering a unique library of genetic material, novel enzymes, and previously unknown chemical compounds that could revolutionize medicine, biotechnology, and our understanding of life's origins. Every sample brought back from the abyss, every new species identified, represents a piece of a puzzle, a key to unlocking the planet's deepest secrets. Yet, despite its critical importance and compelling mysteries, the deep ocean remains largely out of sight, and therefore often out of mind. It is a realm we have only just begun to truly explore. Thanks to incredible advancements in marine technology, from remotely operated vehicles, or ROVs, to autonomous underwater vehicles, AUVs, and highly specialized submersibles capable of withstanding the immense pressures. Each expedition pushes the boundaries of our knowledge, bringing back stunning images and data that continually reshape our perception of this vast, hidden world. So, as we embark on this exploration, prepare to suspend your preconceived notions. Prepare to be amazed by the resilience of life in the most extreme conditions imaginable. Prepare to delve into ecosystems powered not by the sun's warmth, but by the Earth's own internal heat. And prepare to confront the profound beauty and fragility of a frontier that holds the keys to understanding our planet's past, present, and future. The lure of the abyss calls to us not just for its wonder, but for the profound lessons it holds about life, adaptation, and our place within the grand, intricate tapestry of the natural world. This is an invitation to explore Earth's last true wilderness, a journey into the deep ocean and its uncharted mysteries. Our exploration of the deep ocean begins by first understanding how marine biologists define its various realms, moving downwards from the surface. The ocean isn't a uniform body of water. It's a stratified environment, much like layers in an atmosphere, with distinct physical and biological characteristics dictated primarily by the penetration of sunlight. This stratification allows us to delineate different zones, each presenting its own unique set of challenges and opportunities for life. At the very top, from the surface down to approximately 200 meters, we have what is known as the epipelagic zone, often referred to as the sunlit zone. This is the realm most familiar to us. Here, sunlight penetrates sufficiently to support photosynthesis, the process by which marine plants and microscopic phytoplankton convert light energy into organic matter. This abundant sunlight fuels the base of nearly all marine food webs. Think of the vibrant coral reefs, the teeming schools of fish, the majestic whales and dolphins, and the vast blooms of plankton that give the ocean its fertile, shimmering surface. The epipelagic zone is warm, well-mixed, and rich in oxygen, creating a hospitable environment for the vast majority of oceanic life we typically envision. It is a world bursting with color and activity, driven by the energy of the sun. However, as we descend beyond the 200-meter mark, a profound transformation begins. The light, which was once so bright and life-giving, starts to diminish rapidly. The temperature begins its slow, steady descent, and the pressure of the water column above starts to mount significantly. This transition ushers us into our first true segment of the deep ocean, the mesopelagic zone. The mesopelagic zone extends from about 200 meters down to 0 meters. Its name itself, derived from Greek, tells us something fundamental. Meso, meaning middle, and pelagic, referring to the open ocean. But this zone is perhaps more evocatively known as the twilight zone. And for good reason. Here, sunlight is still present, but it's incredibly dim, filtered, and diffused, resembling the muted light of dusk or dawn rather than full daylight. Crucially, there isn't enough light for photosynthesis to occur. This means that unlike the sunlit zone above, life in the twilight zone cannot rely on primary production from plants. It must find its energy elsewhere. The quality of light in the mesopelagic zone is also noteworthy. As light penetrates water, different wavelengths are absorbed at different rates. Red light is absorbed first, then yellow, and finally green. Blue light, with its shorter wavelength, penetrates the deepest. This is why when you look into the deep ocean, everything often appears in shades of blue and gray. For creatures living here, their eyes are adapted to detect these subtle blue hues. The physical conditions in the mesopelagic are challenging. Temperatures typically range from about 20 degrees Celsius at the top down to as low as 4 degrees Celsius at its bottom boundary, making it considerably colder and more stable than the sunlit layer. And then there's the pressure. At 200 meters, the pressure is about 20 times that at the surface. By the time you reach 0 meters, it's 100 times atmospheric pressure, equivalent to having a large car pressing down on every square inch of your body. Imagine that immense force. It's a testament to the incredible engineering of life, that organisms can not only survive, but thrive under such conditions. Life in the mesopelagic zone is a masterclass in adaptation to low light and increasingly scarce food. Without photosynthesis, creatures here must rely on detritus, the rain of organic matter sinking from the productive surface waters, known as marine snow, or by preying on other organisms. One of the most fascinating and widespread adaptations is the evolution of remarkably large eyes. Many mesopelagic fish, like the famous barrel-eye fish or various species of squid, possess tubular, upward-facing eyes designed to capture every last photon of dim sunlight or the fleeting flashes of bioluminescence from other creatures. Another prevalent adaptation is bioluminescence, the ability to produce light chemically. In the twilight zone, bioluminescence isn't just a spectacle, it's a fundamental survival tool. Many mesopelagic organisms use counterillumination, producing light from their undersides to match the dim light filtering down from above, effectively eliminating their shadow and making them invisible to predators looking up. Others use flashes of light for communication, to startle predators, or to lure prey, like the famous anglerfish, whose glowing lure can be found in deeper parts of the mesopelagic and beyond. The mesopelagic is truly where bioluminescence becomes pervasive and crucial. Perhaps the most dramatic display of life in the mesopelagic is the vertical migration phenomenon. Each night, billions upon billions of organisms, from small crustaceans like krill to vast schools of lanternfish, which are considered the most abundant vertebrates on Earth, undertake the largest animal migration on our planet. They ascend from the safety of the twilight zone into the nutrient-rich sunlit layer to feed under the cover of darkness, avoiding predators that rely on vision. As dawn approaches, they descend back into the relative safety and concealment of the mesopelagic. This massive daily movement is so extensive that it forms a deep scattering layer that can actually be detected by sonar, often mistaken for a false seafloor. Other notable residents of the twilight zone include the fearsome-looking viperfish, with its impressive fangs, and the delicate hatchetfish, with their distinctive deep bodies and upward-pointing eyes, perfectly adapted for counterillumination. There are also numerous species of gelatinous zooplankton, like siphonophores and various types of jellyfish, whose fragile, transparent bodies are perfectly suited to the low-light, low-food environment. The mesopelagic zone, then, is a critical bridge. It connects the vibrant, sun-drenched surface world with the truly abyssal depths that lie beneath. It acts as a massive processing plant, playing a crucial role in the global carbon cycle by transferring organic matter from the surface to the deep through the daily migrations and the continuous rain of marine snow. Understanding this twilight world is essential for comprehending the entire oceanic ecosystem, as it is a zone of transition, adaptation, and immense, largely unseen biological activity. It sets the stage for the even more extreme conditions we will encounter as we plunge further into the perpetual night of the deep ocean. Having explored the sunlit epipelagic and the dimly lit twilight zone, the mesopelagic, we now plunge deeper, beyond the reach of even the faintest glimmer of sunlight, into realms of perpetual night and immense pressure. This is where the true deep ocean begins, a world fundamentally different from anything we experience on the surface. Our next descent takes us into the bathypelagic zone, spanning from approximately 1.0 meters down to 4.0 meters. This is famously known as the midnight zone. Here, the concept of light, as we know it, ceases to exist. No sunlight penetrates this far. The only illumination is that which organisms themselves produce, the ethereal glow of bioluminescence, or the occasional flash from specialized research submersibles. Imagine a world where every flicker of light is a living signal, a deliberate act of creation in an otherwise boundless void. The physical conditions in the midnight zone are relentlessly consistent and extreme. Temperatures hover just above freezing, typically between 2 to 4 degrees Celsius, a chilling constancy maintained by the vast volume of water. But it is the pressure that truly defines this zone. At 1.0 meters, you are already under 100 times the atmospheric pressure at sea level. By 4.0 meters, this pressure quadruples to an astonishing 400 atmospheres. To put that into perspective, imagine the weight of 400 full-sized cars stacked on your head. Organisms living here are subjected to forces that would instantly crush any surface creature, collapsing bones and rupturing cells. How does life survive, let alone thrive, under such conditions? The adaptations are profound. Unlike the mesopelagic, where some creatures still possess large eyes to detect faint light or counter-illumination, vision in the bathypelagic often becomes less crucial. Many creatures have greatly reduced eyes, or are entirely blind, relying instead on other senses like chemoreception, an acute sense of smell and taste, or mechanorception, detecting vibrations in the water. For those that still use vision, their eyes are often adapted to detect only specific wavelengths of bioluminescent light, often blue or green, produced by other deep-sea inhabitants. Food scarcity is another dominant challenge. With no sunlight, there are no photosynthetic primary producers. Life in the midnight zone is entirely dependent on food raining down from the productive surface waters, the marine snow, or on preying upon other deep-sea creatures. This scarcity means that organisms must be incredibly efficient with their energy. Many have very slow metabolisms, growing slowly and living for extended periods. Their bodies are often soft and gelatinous, reducing the amount of energy needed for skeletal support. Muscles are often poorly developed, designed for slow, deliberate movements rather than bursts of speed. Reproduction in the bathypelagic also presents unique challenges. Finding a mate in a vast, dark, sparsely populated environment is incredibly difficult. Evolution has devised ingenious solutions. Some male anglerfish, for example, are much smaller than females, and upon encountering a female, will permanently fuse with her, becoming a parasitic sperm donor for the rest of his life. Other species use powerful pheromones to attract mates across great distances, or elaborate bioluminescent displays, unique to their species, as mating signals. Iconic residents of the midnight zone include the anglerfish, famous for its bioluminescent lure, a modified fin ray that dangles like a fishing rod to attract unsuspecting prey. There are also the gulper eels, with their enormous mouths that can engulf prey much larger than themselves, an adaptation for rare feeding opportunities. Various species of snailfish hold the record for some of the deepest living fish, often found in trenches at depths well beyond 4.0 meters. Their soft, gelatinous bodies perfectly suited to the intense pressure. Deep-sea squid and octopuses, often with large eyes or unique bioluminescent capabilities, also navigate this dark realm. As we descend past 4.0 meters, we enter the abyssal pelagic zone, commonly known as the abyssal zone. This vast area extends from 4.0 meters down to 6.0 meters, covering the expansive, flat abyssal plains that make up the majority of the deep ocean floor. This is Earth's largest single habitat, covering over half of the planet's surface. Conditions here are even more stable and extreme than in the bathypelagic. Temperatures hover just above freezing, often around 1 to 2 degrees Celsius. The pressure is truly immense, ranging from 400 to 600 atmospheres. Yet, life persists. The abyssal plains are often covered in fine sediments, composed of the accumulated remains of marine snow that has finally settled after its long descent. Life in the abyssal zone is characterized by creatures adapted to feeding on this sparse rain of detritus. Deposit feeders like sea cucumbers, brittle stars, and various worms are abundant, sifting through the sediments for organic particles. Scavengers such as amphipods, resembling giant versions of tiny shrimp, quickly home in on larger food falls like dead whales or fish, which provide temporary oases of abundance. These creatures are often incredibly resilient, capable of withstanding the crushing pressures and periods of extreme starvation. Their metabolisms are even slower than those in the midnight zone, and their lifespans can be extraordinarily long. While biomass, the total weight of living organisms, is generally low in the abyssal zone, the biodiversity can actually be surprisingly high in certain areas. Scientists are still discovering new species with every expedition to these remote plains, highlighting the hidden richness of this seemingly desolate environment. Many of these organisms are small, often less than an inch long, and possess remarkably delicate or transparent bodies. The transition from the sunlit surface to the abyssal depths is a journey from abundance and warmth to scarcity and cold, from light to eternal darkness. Each zone presents increasingly formidable challenges, yet life has found a way to not just survive, but to flourish in astonishing and unexpected forms. Understanding the characteristics of the bathypelagic and abyssal pelagic zones provides the essential context for appreciating the truly alien environments of the deep, and for marveling at the incredible tenacity and adaptability of life on Earth. These zones are not just cold, dark voids. They are dynamic, albeit slow motion, ecosystems, playing a crucial role in global nutrient cycling and holding a vast, unexplored library of biological wonders. Our journey into the abyssal depths has brought us to the vast plains of the abyssal pelagic zone. But there is one final, even more extreme frontier within the deep ocean, a place of ultimate depth and crushing pressures: the Hadal Zone. Named after Hades, the Greek god of the underworld, this zone encompasses the deepest parts of the ocean found within oceanic trenches and troughs. These are not merely deep areas. They are geological chasms, often V-shaped, formed by the subduction of one tectonic plate beneath another. The Hadal Zone begins at depths greater than 6.0 meters and extends to the very deepest points of the ocean, which can exceed 11.0 meters. There are about 24 known oceanic trenches globally, with the vast majority found in the Pacific Ocean. The most famous and deepest of these is the Mariana Trench in the western Pacific, where the Challenger Deep reaches an astonishing 10,929 meters, or nearly 7 miles down. To put that into perspective, if Mount Everest, the world's highest peak, were placed in the Challenger Deep, its summit would still be more than a mile below the surface of the ocean. These trenches represent the absolute limits of the deep-sea environment. The conditions in the Hadal Zone are beyond extreme. Temperatures remain consistently low, just above freezing, around 1 to 2 degrees Celsius. But it is the pressure that truly sets this zone apart. At 6.0 meters, the pressure is 600 times that at the surface. At the bottom of the Mariana Trench, the pressure exceeds 100 atmospheres, that's over 16.0 pounds per square inch. Imagine the weight of 50 Boeing 747 jumbo jets stacked on top of you. It's a force so immense that it compresses the very water itself, making it slightly denser. Any air-filled spaces, like a human lung, would instantly collapse. For decades, scientists believed that life might not be possible under such unimaginable pressures. The conventional wisdom was that proteins would denature, cellular structures would disintegrate, and metabolic processes would simply cease. However, as technology advanced and we gained the capability to explore these trenches, a startling reality emerged. Life not only exists in the Hadal Zone, but it thrives. While biodiversity might be lower than in shallower depths, the density of certain populations in some trenches can be surprisingly high, forming unique and isolated biological communities. How do hadal organisms cope with these extraordinary pressures? Their adaptations are remarkable. Many hadal creatures exhibit pisophilia, meaning they are pressure-loving or pressure-tolerant. Their cell membranes and proteins have evolved unique structures that prevent them from denaturing or becoming rigid under pressure. Some organisms produce specialized molecules called piezolites, such as trimethylamine N-oxide or TMAO, which stabilize proteins and counteract the effects of high pressure on cellular functions. Their bodies are typically soft-bodied and gelatinous, lacking air bladders or rigid skeletons that would be crushed. Food sources in the hadal zone are even scarcer than in the abyssal plains. Life here is almost entirely dependent on marine snow and larger food falls, like dead fish or whales, that drift down from above and accumulate in these deep depressions. Trenches, acting as natural traps, can concentrate organic matter, creating localized hotspots of food availability. This reliance on external food sources means that the communities found in different trenches, often separated by vast expanses of abyssal plain, can be quite distinct from one another, reflecting the unique organic matter that settles there. Iconic examples of hadal life include various species of amphipods, particularly the gigantic, supergiant amphipods that can grow to over 30 centimeters in length, far larger than their shallow water relatives. These scavengers are incredibly efficient at consuming food falls. Hadal snailfish family Liparidae are another common resident, holding the record for the deepest living fish ever recorded, found at depths exceeding 8.0 meters, and even recently filmed at nearly 340 meters in the Izuagasawara Trench. These fish are remarkably delicate-looking, with translucent bodies and no scales, perfectly adapted to their high-pressure environment. Other life forms include various species of sea cucumbers, holothurians, brittle stars, and highly specialized worms, each uniquely evolved to withstand the extreme conditions. The hadal zone represents the cutting edge of deep-sea exploration and scientific discovery. Each expedition to these trenches uncovers new species and new insights into the limits of life. These isolated environments are natural laboratories for studying evolution under extreme pressure, and for understanding how life can adapt to seemingly insurmountable challenges. The hadal zone is not just the deepest part of the ocean. It is a profound testament to the resilience and adaptability of life on Earth, a final, awe-inspiring frontier in our planet's hidden depths. Having charted the distinct zones of the deep ocean, from the twilight of the mesopelagic to the ultimate depths of the hadal trenches, it's crucial now to delve into the fundamental physical challenges that define these realms. Two of the most immediate and profound obstacles for life in the deep sea are the crushing pressure and the icy, cold temperatures. These aren't just uncomfortable conditions. They are forces that fundamentally reshape the very chemistry and biology of life, demanding extraordinary adaptations. Let's begin with pressure, arguably the most dominant physical characteristic of the deep ocean. As we descend into the water column, the weight of the water above increases dramatically. For every 10 meters, or approximately 33 feet, you go down, the pressure increases by about 1 atmosphere, which is equivalent to the normal air pressure at sea level. This may not sound like much, but consider the cumulative effect. At 1.0 meters in the midnight zone, the pressure is 100 atmospheres. At 4.0 meters, it's 400 atmospheres. And at the bottom of the Mariana Trench, over 900 meters deep, the pressure exceeds an astonishing 100 atmospheres. To truly grasp this, imagine that 1 atmosphere of pressure is roughly equivalent to a kilogram of weight pressing down on every square centimeter of surface area, or about 14.7 pounds per square inch. So, at 100 atmospheres, we are talking about over 16.0 pounds pressing on every square inch. A human body, with its air-filled lungs and cavities, would be instantly crushed beyond recognition at even a fraction of these depths. Any material with air spaces, like wood, would be compressed until it became waterlogged and sank. Even seemingly solid objects can be deformed. For marine organisms, this immense pressure poses a myriad of challenges at every biological level, from the macroscopic to the molecular. Cells, the fundamental units of life, are composed primarily of water, which is largely incompressible. However, the delicate balance of biochemical reactions within those cells, vital for metabolism, growth, and reproduction, is highly sensitive to pressure. High pressure can disrupt the structure and function of proteins, which are the workhorses of every cell. It can alter enzyme activity, preventing crucial chemical reactions from occurring efficiently. It can also affect the fluidity of cell membranes, making them too rigid and hindering essential transport processes. So, how do deep-sea creatures survive without being pulverized? They employ a suite of remarkable adaptations. Firstly, many deep-sea organisms are soft-bodied and lack gas-filled swim bladders, which would simply collapse under pressure. Instead, they rely on neutral buoyancy, achieved through high water content in their tissues, often resembling gelatinous blobs. This means there's no significant pressure differential between the inside and outside of their bodies. They are essentially water, living within water, so the pressure is evenly distributed across their bodies, reducing internal stress. Think of a deep-sea snailfish or a gulper eel, whose flabby, almost translucent bodies are perfectly suited to these conditions. Secondly, at the molecular level, deep-sea organisms have evolved unique biochemical strategies. Their proteins and enzymes are specifically adapted to function optimally under high pressure, sometimes even requiring high pressure to maintain their correct three-dimensional structure. They often produce specialized organic compounds called piezolites, such as trimethylamine N-oxide, or TMAO, and betaines. These piezolites act as molecular chaperones, stabilizing proteins and counteracting the destabilizing effects of pressure on enzyme activity. The deeper an organism lives, the higher the concentration of these piezolites tends to be within its cells, a clear physiological signature of pressure adaptation. Now, let's turn our attention to the other pervasive challenge: icy cold temperatures. Below the sunlit layer, the ocean experiences a dramatic drop in temperature. In the mesopelagic, temperatures typically range from 20 degrees Celsius at the top to about 4 degrees Celsius at its lower boundary. Once you descend into the bathopelagic, abyssal, and hadal zones, the temperature remains remarkably constant, hovering just above freezing, typically between 1 to 4 degrees Celsius. This is because the deep ocean is fed by cold, dense water sinking from the polar regions, creating a vast, cold, stable environment. This near-freezing temperature, coupled with immense pressure, presents its own set of biological hurdles. Low temperatures slow down biochemical reactions, which means that metabolic processes, such as digestion, respiration, and growth, occur at a much slower rate. This explains why many deep-sea creatures exhibit slow growth rates, extended lifespans, and reduced metabolic activity compared to their shallow water counterparts. A deep-sea fish might take decades to reach maturity and live for hundreds of years, far outliving similar species in warmer, shallower waters. To combat the cold, deep-sea organisms also employ various adaptations. Some have evolved specific enzymes that are highly efficient at catalyzing reactions, even at low temperatures. Others accumulate cold-stable lipids fats that remain fluid and functional in their cell membranes, preventing them from becoming rigid and dysfunctional. The absence of extreme temperature fluctuations, however, provides a degree of stability. Unlike surface waters that experience seasonal and daily temperature shifts, the deep ocean is thermally homogeneous, allowing organisms to specialize and optimize for a narrow temperature range. The combination of crushing pressure and icy cold is not merely additive. It creates a complex interplay of stresses. For instance, cold can exacerbate the effects of pressure on protein stability, making the need for piezolytes and specialized enzymes even more critical. Organisms must evolve a harmonious suite of adaptations to simultaneously combat both forces. Consider the unique forms that arise from these pressures. The soft, gelatinous bodies of many deep-sea fish like the snailfish, or the highly flexible skeletons of others. These physical attributes are direct consequences of surviving an environment that would flatten a submarine if it weren't built with extraordinary strength. The lack of air bladders is not just about avoiding collapse. It's about not expending precious energy to maintain buoyancy against incredible downward force. In essence, the deep ocean forces life to operate under conditions that fundamentally rewrite the rules of biological existence. The crushing pressure dictates the architecture of bodies and the chemistry of cells, while the icy cold governs the pace of life. These two omnipresent physical challenges are the architects of the deep-sea fauna, shaping their forms, their metabolisms, and their very survival in a world that is as alien as it is awe-inspiring. Having explored the profound physical challenges of crushing pressure and icy cold, we now turn to two equally defining characteristics of the deep ocean: perpetual darkness and extreme food scarcity. These conditions are inextricably linked and fundamentally shape the adaptations, behaviors, and very existence of life in the abyss. Let's first consider the challenge of perpetual darkness. Below the mesopelagic zone, roughly 1.0 meters down, sunlight ceases to penetrate entirely. This means that for the vast majority of the ocean's volume, there is no light for photosynthesis, the process that fuels almost all life on Earth. Imagine a world where day and night hold no meaning, where the concept of a sunrise or sunset is utterly alien. In this boundless void, the only light produced is by the organisms themselves, through the incredible phenomenon of bioluminescence, which we will explore in detail shortly. The absence of sunlight has profound implications. Without light, there are no plants or phytoplankton to form the base of a food web. This eliminates the primary source of energy that sustains surface ecosystems. Consequently, the deep sea is a world of incredible visual adaptations. or the complete lack thereof. In the upper mesopelagic, where some faint, blue light still filters down, animals often have exceptionally large, sensitive eyes, often tubular, and directed upwards to capture every last photon. Examples include the barrel-eye fish, with its unique, upward-pointing, rotatable eyes, or various deep-sea squid. Their vision is tuned to the blue wavelength, the only color that penetrates to those depths. However, as we descend into the bathypelagic and abyssal zones, where absolute darkness reigns, eyes often become much smaller or are entirely absent. Why maintain a costly sensory organ that has no external light source to perceive? Instead, deep-sea creatures develop highly refined alternative senses. Chemoreception, akin to an incredibly acute sense of smell or taste, becomes paramount. Many deep-sea fish and invertebrates have enhanced olfactory organs to detect the faint chemical trails of prey, predators, or potential mates in the vast darkness. Similarly, mechanoreception, the ability to detect vibrations and pressure changes in the water, is highly developed. The lateral line systems of deep-sea fish, for instance, are exceptionally sensitive, allowing them to navigate, find prey, and avoid collisions in total darkness. Some creatures also employ sophisticated sonar-like systems, emitting and detecting sound waves to map their surroundings. The other inseparable challenge of the deep is extreme food scarcity. This is a direct consequence of the lack of light for photosynthesis. Without primary producers at depth, the entire deep-sea ecosystem is fundamentally dependent on food originating from the sunlit surface waters. This food arrives in a continuous, albeit sparse, rain of organic detritus, famously known as marine snow. Marine snow consists of dead phytoplankton and zooplankton, fecal pellets, discarded larval cases, mucus, and other organic debris, slowly sinking from the productive upper layers. The journey for this marine snow is long and arduous. As it descends, it is constantly being consumed and broken down by bacteria and other organisms, meaning that only a tiny fraction of the surface's productivity ever reaches the abyssal plains and trenches. Estimates suggest that less than 5% of the organic matter produced in the euphotic zone ever makes it past 1.0 meters. This scarcity dictates that every feeding opportunity is critical, and energy conservation is paramount. To cope with this perpetual famine, deep-sea organisms have evolved a wide array of fascinating and often grotesque feeding strategies. Sit-and-wait predators. Many deep-sea fish are ambush predators, equipped with enormous mouths and distensible stomachs, capable of swallowing prey much larger than themselves. The iconic anglerfish, for instance, uses its bioluminescent lure to attract unsuspecting prey to its massive, fanged jaws. The gulper eel, with its disproportionately large mouth and expandable throat, can literally swallow prey as large as itself. These adaptations maximize the yield from infrequent feeding opportunities. Scavengers. A significant portion of the deep-sea fauna are scavengers, highly adapted to locating and consuming sporadic, large food falls. These whale falls, fish falls, or other carcasses provide ephemeral oases of abundance in the otherwise barren landscape. Deep-sea amphipods, resembling giant shrimp, are renowned for their ability to quickly swarm and consume large carcasses, playing a crucial role in recycling nutrients. Their chemoreceptors are incredibly sensitive, allowing them to detect a food fall from miles away. Deposit feeders. On the abyssal plains, where marine snow accumulates as fine sediment, deposit feeders are common. Organisms like sea cucumbers, brittle stars, and various worms simply ingest the sediment, extracting the organic particles and bacteria within it. These creatures act like living vacuum cleaners, processing vast amounts of the seafloor to extract minimal nutrition. Their slow metabolisms are perfectly suited for this low-energy diet. Parasitism and commensalism. Given the difficulty of finding mates and food, some deep-sea species resort to parasitic or symbiotic relationships to secure energy or reproductive opportunities. The male parasitic anglerfish, fusing with the female, is a prime example of an adaptation for reproduction, but it also means the female provides sustenance. Beyond specialized feeding mechanisms, the general physiological adaptations to food scarcity are also profound. Deep-sea animals typically exhibit slow metabolisms, requiring less energy to sustain themselves. This means slower growth rates and significantly longer lifespans. Many deep-sea fish can live for decades, even centuries, growing slowly and maturing late. Their movements are often slow and deliberate, conserving energy, as bursts of speed would be metabolically too expensive. Their bodies are frequently soft and gelatinous, reducing the amount of energy needed for skeletal structures. The constant battle against darkness and hunger has thus shaped the deep-sea ecosystem into a realm of incredible efficiency, ingenuity, and extreme specialization. Every flicker of light, every scent, every vibration, and every morsel of food is a precious commodity. It's a testament to life's profound adaptability that despite these formidable challenges, the deep ocean supports a rich, albeit often sparsely distributed, tapestry of unique and fascinating organisms. Having established the profound challenges of perpetual darkness and food scarcity, we now turn our attention to one of the most astonishing and widespread adaptations found in the deep ocean: bioluminescence. In a world devoid of sunlight, the ability of living organisms to produce their own light is not merely a beautiful spectacle. It is a fundamental, multifaceted tool for survival, communication, hunting, and defense. Bioluminescence is so pervasive in the deep sea that it's estimated over 90 of deep-sea organisms are capable of producing light. If you could see the deep ocean as its inhabitants do, it would be a breathtaking display of flashing, shimmering, and glowing forms. So what exactly is bioluminescence? It is, at its core, a chemical reaction that produces cold light, meaning very little heat is generated. Unlike incandescent light bulbs, which produce light by heating a filament, bioluminescence is an incredibly efficient process. The basic reaction involves two key molecules: a light-emitting compound called luciferin and an enzyme called luciferase. When luciferin is oxidized in the presence of luciferase, often with cofactors like ATP and oxygen, light is produced. Different organisms have evolved different forms of luciferin and luciferase, leading to a dazzling array of colors though predominantly blue and green, in the deep sea, as these wavelengths travel furthest in water and flash patterns. This light can be produced in several ways. Some organisms have specialized light-producing cells called photocytes within their own bodies, which they can control. Others host symbiotic bacteria, typically Vibrio bacteria, within specialized organs called photophores. These photophores can be incredibly complex structures, sometimes equipped with reflectors, lenses, and filters, allowing the organism to precisely control the intensity, direction, and color of the light emitted. The ability to switch the light on and off, or even change its intensity, is often crucial for its effectiveness. The evolutionary drivers for bioluminescence in the deep ocean are numerous and diverse, reflecting the constant selective pressures of darkness and the need to interact with other organisms. Let's explore some of the primary uses. One of the most widespread and critical uses of bioluminescence is counterillumination. This strategy is particularly common in the mesopelagic, or twilight zone, where some faint blue light still filters down from the surface. Many animals here possess photophores on their undersides. By producing light that precisely matches the intensity and color of the dim ambient light filtering from above, these creatures effectively eliminate their own shadow. This makes them virtually invisible to predators or prey looking up from below. Imagine trying to spot a fish against a faint glow from above. Without a shadow, it simply blends into the background. Species like the hatchetfish are masters of counterillumination, with rows of ventral photophores carefully tuned to mimic the downwelling light. Another crucial function is luring prey. In a food-scarce environment, attracting food is a matter of survival. The most famous example of this is the anglerfish. Female anglerfish possess a modified dorsal fin ray that extends forward over their mouths, tipped with a fleshy esca that contains bioluminescent bacteria. This esca acts as a glowing lure, tempting small fish or crustaceans to come closer, only to be snapped up by the anglerfish's formidable jaws. There are many variations on this theme. Some deep-sea squid use bioluminescent tentacles to attract prey, while certain jellyfish produce glowing fishing lines that ensnare unsuspecting organisms. The light here acts as a beacon in the darkness, drawing sustenance directly to the predator. Bioluminescence is also a potent tool for evading predators. When threatened, deep-sea organisms can deploy light in various defensive ways. Burglar alarm effect. Imagine a small copepod being attacked by a fish. The copepod might flash brightly, attracting an even larger predator to the scene. The original predator might then become the prey of the larger animal, allowing the copepod to escape. It's a risky, but often effective, diversionary tactic. Some deep-sea squids, instead of releasing ink like their shallow water cousins, eject a cloud of bioluminescent fluid, creating a dazzling, confusing smokescreen that allows them to make a quick getaway. Startle response. A sudden, bright flash of light can momentarily startle or blind a predator, giving the prey just enough time to escape. Many comb jellies and some crustaceans employ this strategy, producing brilliant, unexpected bursts of light. Sacrificial luminescence. Some organisms can detach a bioluminescent body part, like a glowing tentacle or a glowing scales, to distract a predator, while the rest of the animal escapes into the darkness. This is similar to how a lizard might shed its tail. Finally, bioluminescence is fundamental for communication and mate attraction. In the vast, dark expanse of the deep sea, finding a mate can be incredibly challenging. Species-specific patterns of bioluminescent flashes, unique colors, or even complex light shows serve as critical signals for individuals of the same species. Male and female members of the same species might recognize each other by these distinct light displays, preventing interspecies breeding and ensuring reproductive success. It's a visual language in a world where sound and chemical signals also play important roles, but where light offers an immediate, striking identifier. For instance, some ostracods, tiny crustaceans, perform elaborate bioluminescent dance routines to attract mates. The sheer diversity of bioluminescent applications underscores its evolutionary importance in the deep ocean. It is not just a singular adaptation, but a vast toolkit of solutions to the challenges of living in perpetual darkness. From camouflage to predation, defense to reproduction, bioluminescence transforms the silent, lightless void into a vibrant, flashing, and signaling world, making it one of the most mesmerizing and vital phenomena in the abyssal depths. In our previous discussion, we introduced the fundamental chemical process of bioluminescence and its primary roles in the deep sea. Counterillumination, luring prey, and evading predators through various alarm signals. Now, let's dive deeper into specific examples and more nuanced applications of this incredible light production, further illuminating its critical importance in the abyss. One of the most remarkable examples of bioluminescence for predation is seen in the dragonfish. Unlike the anglerfish, which uses a lure, many species of dragonfish have a unique adaptation: a suborbital photophore that emits red light. Why red light, when blue-green light penetrates deepest and is the most common color produced by other bioluminescent organisms? Because most deep-sea creatures, with eyes adapted to detect blue light, are essentially blind to red light. This gives the dragonfish a significant advantage, acting as a secret weapon. It can effectively shine a red spotlight on its unsuspecting prey, illuminating them without being seen itself. It's like having night vision goggles in a world of normal vision. This stealthy hunting strategy highlights the intricate arms race of light and vision in the deep. Beyond the well-known anglerfish, many other deep-sea fish employ various forms of lures. Some fish have photophores within their mouths, creating a glowing cavern to draw prey directly into their jaws. Others, like the lucijaw fish, can use a string of photophores along their bodies, or even on their detached lure-like filaments, creating a fishing line effect. These different lure designs and positions demonstrate the vast array of evolutionary experiments in attracting food in the dark. The defensive uses of bioluminescence are equally sophisticated. Consider the vampire squid Vampiroteuthis infernalis, a truly unique cephalopod that lives in the oxygen minimum zone, a region of the ocean with extremely low oxygen levels. When threatened, instead of releasing ink like other squid, the vampire squid pulls its arms over its head, essentially turning itself inside out, revealing a spiny, cape-like webbing. From the tips of its arms, it can eject a cloud of bioluminescent mucus, creating a dazzling, disorienting display that acts as a fireworks show. This allows it to quickly escape into the darkness, leaving the predator bewildered by the sparkling distraction. Another fascinating defensive tactic is employed by some species of pyrosums. These are colonial tunicates, forming large, cylindrical colonies that can be several meters long. When disturbed, the entire colony can light up in a brilliant, undulating glow. This collective display can be overwhelming for a predator, making the colony appear much larger and more formidable, or simply startling enough to deter an attack. The role of bioluminescence in communication and reproduction is subtly complex. While we mentioned species-specific flash patterns, it's worth noting the variety. Some ostracods, tiny crustaceans, engage in elaborate, species-specific courtship displays, flashing light in intricate sequences that are unique to their kind, ensuring that mates recognize each other in the dark. These patterns can involve precise timing, duration, and spatial arrangements of light. Other organisms might use a steady glow or a particular color to signal their presence or readiness to mate. The ability to precisely control the light, turning it on and off, varying its intensity, or even changing its color, is a testament to the sophisticated neural and muscular control mechanisms that have evolved. Beyond active light production, some deep-sea organisms utilize bioluminescence in more passive ways. For instance, the transparent nature of many deep-sea creatures, particularly gelatinous zooplankton, helps them avoid detection by predators using bioluminescent searchlights. If light passes straight through you, you cast no shadow and are effectively invisible even when illuminated. The study of bioluminescence also has profound implications for our understanding of fundamental biological processes, and even for potential technological applications. The enzymes luciferases and light-emitting compounds luciferins involved in bioluminescent reactions are incredibly efficient and specific. Scientists are studying these systems for a variety of uses, from developing new biosensors to detect pollutants, to creating new medical diagnostic tools, and even for generating light in an energy-efficient manner. The stability of these compounds under extreme pressure and cold also offers insights into protein engineering and the limits of molecular function. The prevalence of bioluminescence also gives us a unique window into the evolutionary history of life. The ability to produce light has evolved independently many times across diverse groups of organisms, from bacteria and fungi to jellyfish, fish, and squid. This convergent evolution underscores the immense selective pressure and undeniable advantage that light production provides in a perpetually dark environment. It suggests that once life descended into the aphotic zone, the ability to generate light became a powerful currency for survival and interaction. In summary, bioluminescence is far more than just a captivating phenomenon. It is the language of the deep. It dictates who sees whom, who eats whom, who finds a mate, and who survives. It transforms the vast, dark emptiness into a dynamic, pulsating world of living light, a testament to the ingenious ways life, adapts to even the most extreme conditions on Earth. This internal light source is the key to unlocking the mysteries of interaction and survival in the deep ocean's uncharted realms. We've previously discussed the profound physical challenges of the deep ocean: the crushing pressure, the icy cold, perpetual darkness, and extreme food scarcity. Now, let's explore how deep-sea organisms have evolved truly remarkable physiological and anatomical adaptations to not just survive, but often thrive in these seemingly inhospitable conditions. These adaptations touch upon every aspect of their biology, from their basic body structure to the very rate of their life processes. One of the most striking physiological adaptations is related to metabolism. In a world of scarce food and cold temperatures, energy conservation is paramount. Deep-sea organisms generally exhibit incredibly slow metabolic rates. This means their bodily processes, digestion, respiration, growth, and reproduction, all occur at a much slower pace than those of their shallow water counterparts. This allows them to subsist on the sparse nutrients available and endure long periods between meals. For example, some deep-sea fish might take decades to reach maturity and live for hundreds of years. The Greenland shark, an inhabitant of cold, deep waters, can live for over 500 years, making it the longest-living vertebrate on Earth. This slow, deliberate pace of life is a direct evolutionary response to a low-energy environment. This slow metabolism is also reflected in their movement. Many deep-sea fish and invertebrates move very slowly and deliberately. Fast, powerful bursts of speed are metabolically expensive and would quickly deplete their limited energy reserves. Instead, they often rely on sit-and-wait ambush predation or slow, methodical scavenging. Their muscles are typically less developed and have a higher water content, further reducing the energy cost of movement. Related to metabolism are adaptations in body composition. Many deep-sea animals have gelatinous, watery bodies with minimal skeletal structures. Think of the deep-sea snailfish, with its soft, almost translucent appearance, or the various species of comb jellies and siphonophores. This high water content helps in several ways. Firstly, it makes the organism effectively incompressible, as water is largely incompressible. By minimizing air-filled spaces and solid bone muscle mass, there's less material for the immense pressure to crush or deform. Secondly, constructing and maintaining dense, calcified skeletons or muscular bodies requires significant energy, which is a luxury in the deep sea. By adopting a flabby or gelatinous structure, organisms conserve vital energy. This also contributes to neutral buoyancy without the need for an energetically costly swim bladder, which would collapse under pressure. Their skeletal structures, when present, are often remarkably different from surface dwellers. Bones are typically less dense, more cartilaginous, and demineralized. This reduction in mineral content makes them less rigid and more flexible, allowing them to withstand pressure without fracturing. Another key physiological adaptation for pressure survival is at the molecular level. As discussed, high pressure can denature proteins and enzymes. Deep-sea organisms produce specialized organic compounds called piezolites, such as trimethylamine N-oxide TMAO, betaines, and sarcosine. These compounds accumulate in their cells and act as protein stabilizers, counteracting the effects of pressure on protein structure and function. The concentration of TMAO, for instance, generally increases with depth, reaching very high levels in hadal zone organisms like snailfish. These piezolites help maintain the correct three-dimensional shape of proteins, allowing biochemical reactions to proceed efficiently, even under thousands of atmospheres of pressure. Furthermore, the cell membranes of deep-sea organisms are adapted to the cold and pressure. They often have a higher proportion of unsaturated fatty acids, which helps maintain the fluidity and flexibility of the membrane at low temperatures and high pressures, ensuring that vital transport and signaling processes can occur. While extreme conditions generally lead to smaller sizes in some environments, the deep sea also exhibits the phenomenon of deep-sea gigantism. This is where certain invertebrates, such as some species of amphipods, isopods, copepods, and even jellyfish and squid, grow to sizes far exceeding their shallow water relatives. Examples include the giant isopod Bathynomus giganteus, which can reach over 75 cm in length, or the legendary giant squid Architeuthis dux, and colossal squid Mesonychoteuthis hamiltoni, which are among the largest invertebrates on Earth, with eyes the size of soccer balls. The reasons for deep-sea gigantism are thought to be multifactorial. Cold temperatures. Lower temperatures lead to slower metabolic rates. Animals that grow slowly and mature late can continue to grow throughout their long lives, eventually reaching enormous sizes. Reduced predation pressure. While predators exist, the overall density of predators may be lower than in surface waters, allowing some species to grow larger without being as vulnerable to predation. Food scarcity and big meals. For scavengers like the giant amphipods, larger body size might enable them to consume and process large, infrequent food falls more efficiently. A bigger body can store more energy reserves, allowing for longer periods between meals. Pressure effects. Some theories suggest that increased pressure might somehow facilitate larger cell size or growth rates in certain invertebrates, although this is less understood than other factors. Conversely, some deep-sea organisms exhibit dwarfism, particularly certain species of fish and crustaceans. This is often an adaptation to the extreme food scarcity, where a smaller body size requires less energy to maintain, making survival more feasible on a very limited diet. The most successful strategy for size often depends on the specific niche and feeding strategy of the organism. In summary, the deep ocean demands a complete biological overhaul. From their incredibly slow metabolisms and energy-efficient, gelatinous bodies to their pressure-stabilizing molecules and uniquely adapted cellular components, deep-sea creatures are living marvels of evolution. Whether exhibiting gigantism or dwarfism, their physiological and anatomical traits are finely tuned responses to a world defined by immense pressure, frigid cold, perpetual darkness, and the constant threat of starvation. They are not merely surviving. They are demonstrating the astonishing plasticity and resilience of life itself. We have spent considerable time describing the vast, cold, dark, and food-scarce expanse of the deep ocean. where life is largely dependent on the meager rain of marine snow from above. However, there exist extraordinary exceptions to this rule. Vibrant, bustling oases of life, thriving in complete darkness and under immense pressure, powered not by the sun, but by the Earth's own internal heat. These are the hydrothermal vents. Their discovery in 1977 fundamentally revolutionized our understanding of where and how life could exist, challenging long-held assumptions about the necessity of sunlight for ecosystem function. The story of their discovery is as remarkable as the vents themselves. In 1977, aboard the submersible Alvin, scientists were exploring the Galapagos Rift, a segment of the mid-ocean ridge in the Pacific. They were searching for areas of geothermal activity, theorized to exist where tectonic plates pull apart, allowing magma to rise close to the seafloor. What they found was beyond anything they could have imagined. Towering chimney-like structures spewing superheated, mineral-rich fluids, surrounded by dense, flourishing communities of bizarre and previously unknown life forms. This was a paradigm shift in marine biology and astrobiology. So what exactly are hydrothermal vents? They are openings in the seafloor, typically found along mid-ocean ridges, volcanic hot spots, and subduction zones, where Earth's tectonic plates are diverging or converging. Here, seawater seeps down through cracks in the oceanic crust, sometimes several kilometers deep. As this water percolates downwards, it gets heated by the underlying magma chamber, reaching incredibly high temperatures, often exceeding 350 to 400 degrees Celsius, over 700 degrees Fahrenheit. This superheated water undergoes a crucial transformation. It reacts chemically with the surrounding rocks, dissolving metals like iron, copper, zinc, and manganese, as well as picking up hydrogen sulfide and methane. Because of the immense pressure at these depths, the water, despite being far above its boiling point at surface pressure, remains in a liquid state. This hot, chemically altered, and highly acidic fluid then rises back to the seafloor, spewing out of vent openings. As this superheated, chemical-rich fluid meets the cold 2 to 4 degrees Celsius ambient deep-sea water, the dissolved minerals rapidly precipitate out of solution, forming towering structures around the vents. These chimney-like formations can grow several stories high. The most iconic are black smokers, which emit dark, mineral-laden plumes resembling smoke due to the precipitation of iron and sulfide minerals. There are also white smokers, which are typically cooler and emit lighter-colored plumes rich in barium, calcium, and silicon. The geological processes creating these vents are a direct manifestation of plate tectonics and the Earth's dynamic inner workings. The truly revolutionary aspect of hydrothermal vents, however, lies not in their geology, but in their biology. Unlike the vast majority of life on Earth that relies on photosynthesis as the ultimate energy source, vent ecosystems are powered by a process called chemosynthesis. Instead of converting light energy into chemical energy, specialized microorganisms, primarily bacteria and archaea, convert the chemical energy from inorganic compounds, particularly hydrogen sulfide H2S, into organic matter. This process is analogous to photosynthesis in its outcome, but fundamentally different in its energy source. These chemosynthetic microbes form the absolute base of the vent food web. They are the primary producers, transforming the toxic hydrogen sulfide and other chemicals from the vent fluids into usable forms of carbon. Without these chemosynthetic organisms, the entire vent community would not exist. The specific chemosynthetic reaction often involves the oxidation of hydrogen sulfide. H2SO2 CO2 CH2O organic matter S-sulfur H2O. This reaction releases energy, which the microbes then use to fix carbon dioxide and synthesize organic compounds. It's a remarkable testament to the adaptability of life, demonstrating that ecosystems can flourish in the complete absence of sunlight, drawing their vitality directly from the Earth's geological processes. The vibrant communities that arise around these vents are incredibly dense and diverse, a stark contrast to the sparse populations of the surrounding abyssal plains. Imagine standing at the bottom of the ocean in total darkness and coming across a bustling metropolis of life. Giant tube worms, massive clams, unique species of mussels, strange shrimp, and bizarre crabs all congregate around the vents. The life here is so abundant that it can reach biomass levels comparable to tropical rainforests or coral reefs, making them true oases in the deep. These vent communities are often short-lived in geological terms. Vent fields can be active for decades to centuries, but eventually, the flow of superheated fluids can cease as the underlying geological activity shifts. When a vent dies, the community it supports also perishes, highlighting the transient nature of these remarkable deep-sea habitats. However, new vents can form elsewhere along the ridges, and the dispersal of larvae from one vent field to another is a critical area of ongoing research. The discovery of hydrothermal vents changed everything. It opened up entirely new fields of research in deep-sea biology, microbiology, and geology. It provided compelling evidence for the possibility of life existing in extreme environments on other planets or moons, such as Jupiter's moon Europa or Saturn's moon Enceladus, where similar subsurface oceans and hydrothermal activity might exist. It forced us to broaden our definition of life's fundamental requirements and its incredible resilience. The chemosynthetic world of hydrothermal vents remains one of the most astonishing discoveries in marine science, a vivid reminder that the ocean holds wonders beyond our wildest imagination. In our previous segment, we introduced the revolutionary discovery of hydrothermal vents and the concept of chemosynthesis, the process that powers these incredible ecosystems. Now, let's delve deeper into the extraordinary communities that thrive around these vents, focusing on the specific organisms and the intricate symbiotic relationships that define these unique oases of life. The most iconic inhabitants of hydrothermal vents are undoubtedly the giant tube worms, scientifically known as Riftia pachyptilla. These remarkable creatures are unlike any other animals on Earth. They are invertebrates that can grow to over 4 meters 8 feet in length, forming dense thickets around the vent openings. What makes them truly unique is their complete lack of a mouth, digestive tract, or anus as adults. How do they feed? They rely entirely on an extraordinary symbiotic relationship with chemosynthetic bacteria. Inside a specialized organ called the trophosome, located within the tube worm's body, live billions of these bacteria. The tube worm has evolved a highly efficient blood system that delivers oxygen, carbon dioxide, and crucially, hydrogen sulfide directly to these internal symbionts. The bacteria then perform chemosynthesis, converting the hydrogen sulfide into organic carbon compounds, which are then used to nourish the tube worm. In return for a stable environment and a constant supply of chemicals, the bacteria provide all the nutrition the worm needs. The vibrant red plume that extends from the top of the tube worm's tube is called the branchial plume, which is rich in hemoglobin similar to our blood and efficiently absorbs oxygen and hydrogen sulfide from the vent fluids. This stunning example of mutualism highlights how life can forge entirely new biological pathways in extreme environments. Beyond the giant tube worms, other large invertebrates also form the core of these vent communities, often engaging in similar symbiotic relationships. Giant white clams Calyptogena magnifica and giant mussels Bathymodiolus thermophilus are also prominent. Like the tube worms, these bivalves host chemosynthetic bacteria within their gill tissues. They filter the vent fluids, allowing their bacterial partners to metabolize hydrogen sulfide and produce food for their hosts. These clams can grow to extraordinary sizes, sometimes over 30 centimeters 1 foot long, forming vast beds around the vents. Then there are the Pompeii worms, Alvinella pompejana, named after the buried city because they live on the very walls of black smoker chimneys, thriving in one of the most extreme thermal gradients on Earth. One end of their body can be in water as hot as 80 degrees Celsius, 176 F, while the other end is in ambient deep-sea water of just a few degrees Celsius. These worms are covered in a fleece-like layer of bacteria, suggesting another complex symbiotic relationship that may help them insulate or detoxify. Their existence challenges our understanding of the thermal limits of animal life. Vent crabs and vent shrimp are also abundant at hydrothermal vents, while some species, like the blind shrimp Rimicaris hybisi, also host chemosynthetic bacteria internally or on their mouthparts. Many are grazers or predators. They feed on the bacterial mats that carpet the vent surfaces or prey on the smaller invertebrates that thrive on these mats. Some vent crabs, like the hairy-chested crab Kiwa puravita, cultivate bacterial gardens on the bristles of their chests, sweeping them off to eat. Their unique sensory adaptations, often involving highly specialized chemoreceptors, allow them to navigate the turbulent chemical gradients of the vent fields. Fish are also present, though often less prominent than the invertebrate communities. Vent-dwelling fish, like various species of eelpouts Zorsidae and Bythetidae, are typically generalist predators or scavengers, feeding on the rich biomass of worms, crustaceans, and other invertebrates associated with the vents. They are adapted to the unique chemical environment and high pressure, but generally do not possess the same direct symbiotic relationships as the primary consumers. The structure of a hydrothermal vent ecosystem is a classic example of zonation. As you move away from the immediate vent opening, where temperatures are highest and chemical concentrations are most extreme, the species composition changes. Closest to the vent are the most specialized organisms, such as Riftia and Alvanella, which can tolerate or require high concentrations of hydrogen sulfide and extreme temperatures. Further out, where the fluids are more diluted and cooler, you find more generalized consumers and scavengers. The discovery and continued study of hydrothermal vent ecosystems have had a profound impact on several scientific fields. Origin of life. The idea that life might have originated in similar chemical-rich, energy-abundant environments on early Earth rather than in shallow, sunlit ponds has gained significant traction. Hydrothermal vents offer a compelling analogy for primitive Earth conditions. Astrobiology. The existence of these ecosystems provides strong evidence that life could exist on other planetary bodies, such as the ice-covered moons of Jupiter Europa and Saturn Enceladus, which are thought to harbor vast subsurface oceans with potential hydrothermal activity. These findings broaden the search for extraterrestrial life beyond Earth-like conditions. Biodiversity. Vents are biodiversity hotspots containing unique lineages of life found nowhere else. The ongoing discovery of new species continues to reveal the incredible adaptability and evolutionary ingenuity of life in extreme environments. Biotechnology. The extremozymes, enzymes from extremophiles that thrive in high temperatures, pressures, or chemical concentrations found in vent organisms hold immense potential for industrial and medical applications, as they can function under conditions that would destroy conventional enzymes. Hydrothermal vents are not merely curiosities. They are living laboratories demonstrating alternative pathways for life, powered by geological forces rather than solar energy. They are a constant reminder of the ocean's profound capacity to surprise and challenge our assumptions about life on Earth and potentially beyond. These bustling oases in the otherwise stark deep-sea environment continue to reveal the remarkable tenacity and diversity of life. While hydrothermal vents represent the most famous example of chemosynthesis-powered deep-sea ecosystems, they are not the only oases of life in the abyss. The deep ocean harbors other unique habitats, equally reliant on chemical energy or large organic inputs, that create vibrant, albeit localized, biological hotspots. Today, we will explore two such fascinating environments: cold seeps and whale falls. These discoveries further expand our understanding of how life can find and exploit energy sources in the seemingly barren deep-sea floor. Let's begin with cold seeps. As their name suggests, cold seeps are areas on the seafloor where fluids, rich in hydrogen sulfide, methane, and other hydrocarbons, seep out of the ocean floor, often at ambient deep-sea temperatures, hence cold, compared to the superheated hydrothermal vents. These seeps occur in various geological settings, including passive continental margins, subduction zones, and gas hydrate stability zones. Unlike vents, which are typically found along active spreading centers, cold seeps are often associated with areas where buried organic matter decomposes, producing methane, or where oil and gas reserves are present beneath the seafloor. Similar to hydrothermal vents, The base of the food web at cold seeps is chemosynthesis. Here, specialized bacteria and archaea utilize methane, hydrogen sulfide, and other reduced compounds as their energy source, converting them into organic matter. This process, just like at vents, supports a diverse community of larger organisms that either feed directly on these microbes or form symbiotic relationships with them. The communities found at cold seeps share many similarities with vent communities, suggesting convergent evolution in response to chemosynthetic energy sources. Giant clam beds, often Calyptogena or Bathymodiolus species, are incredibly common. These clams house chemosynthetic bacteria within their gills, much like their vent counterparts, absorbing the necessary chemicals from the seeping fluids. Large mussel beds, also hosting chemosynthetic symbionts, are also characteristic features. Tube worms are also prominent at cold seeps, though they belong to different genera than the Riftia found at vents. The most famous cold seep tube worms are from the genus Lamellibranchia. These worms can grow even larger than Riftia, sometimes exceeding 3 meters 10 feet in length, and can live for hundreds of years. Like Riftia, they lack a mouth or digestive system and rely entirely on internal chemosynthetic bacteria. Their long lifespans highlight the slow pace of life and stability of some cold seep environments, which can remain active for thousands of years, much longer than hydrothermal vents. Other organisms found at cold seeps include various species of crabs, shrimp, snails, and specialized fish that prey on the chemosynthetic primary consumers, or on the bacterial mats that often blanket the seafloor around the seeps. The fluids seeping from cold seeps can also contain methane hydrates, often referred to as flammable ice. These are ice-like solids composed of water and methane, formed under high pressure and low temperature. When destabilized, they can release methane gas, further fueling the chemosynthetic communities. Cold seeps are crucial for understanding the diversity of life in the deep sea and the global carbon cycle. They represent significant reservoirs of methane, a potent greenhouse gas, and the chemosynthetic processes at seeps play a role in consuming this methane, preventing its release into the water column or atmosphere. Now let's transition to a truly unique and ephemeral deep-sea habitat, Whale Falls. Imagine the vast, open ocean where food is incredibly scarce. Then, a massive source of organic matter, a dead whale, sinks thousands of meters to the abyssal seafloor. This event, known as a whale fall, creates a sudden, enormous feast, transforming a barren landscape into a thriving, though temporary, ecosystem. The discovery of whale falls as distinct ecosystems was relatively recent, beginning in the late 1980s. Scientists found surprisingly diverse communities of organisms rapidly colonizing these carcasses, highlighting another pathway for energy to reach the deep. A whale fall community progresses through several distinct stages. 1. Mobile scavenger stage. Within weeks to months of a whale sinking, highly mobile scavengers, such as hagfish, sleeper sharks, giant amphipods, and various crabs, are drawn from miles around by the scent of the decaying flesh. They rapidly consume the soft tissues, stripping the carcass down to the bones within months. This stage is characterized by intense feeding frenzies. 2. Enrichment opportunist stage. Once most of the soft tissue is gone, the surrounding sediments become enriched with organic matter and lipids released from the carcass. This stage, lasting months to years, sees an explosion of opportunistic organisms like polychaete worms, small crustaceans, and gastropods. These creatures feed on the remaining scraps of flesh and the bacteria that flourish on the decomposing tissues and enriched sediments. 3. Sulfophilic stage. This is the longest and most fascinating stage, lasting for decades or even a century or more for very large whales. At this point, the bones, which are rich in lipids fats, become the primary energy source. Specialized sulfophilic sulfur-loving bacteria colonize the bones and metabolize the lipids anaerobically, producing hydrogen sulfide as a byproduct. This hydrogen sulfide then fuels a chemosynthetic community, much like at hydrothermal vents and cold seeps. The most iconic organisms of the sulfophilic stage are the bone-eating worms of the genus Ostax. These bizarre worms, aptly nicknamed zombie worms, were only discovered in 2002. They have no mouth or gut. Instead, they bore into the whale bones using root-like structures, through which they absorb lipids and proteins. They host symbiotic bacteria within these roots that help them digest the bone lipids. Female Ostax worms are much larger than males and often contain dwarf males living parasitically inside them. Other organisms found at this stage include specialized limpets, snails, and various invertebrates that graze on the chemosynthetic bacterial mats covering the bones. Whale falls, therefore, demonstrate a truly unique form of chemosynthesis, one that is not driven by geological activity, but by the slow decomposition of massive organic input. They provide crucial stepping stones for the dispersal of chemosynthetic species across the deep-sea floor, acting as temporary islands that connect various vent and seep sites. They also offer valuable insights into the decomposition of organic matter in the deep and the long-term cycling of nutrients. In conclusion, cold seeps and whale falls, alongside hydrothermal vents, powerfully illustrate the diverse and ingenious ways life has colonized the deep ocean. These habitats, fueled by chemical energy rather than sunlight, showcase the remarkable plasticity of life and its ability to thrive in seemingly impossible conditions. They are constant reminders that our planet's deepest realms continue to hold surprises, revealing entirely new paradigms for how ecosystems function. Our journey through the deep ocean has revealed a world of extreme conditions and astonishing life. But how do we, as surface-dwelling humans, access and study these remote and hostile environments? The answer lies in remarkable technological innovations that have progressively pushed the boundaries of deep-sea exploration. It is through the development of specialized platforms like remotely operated vehicles, autonomous underwater vehicles, and manned submersibles that we have been able to peer into the abyss and unveil its secrets. For much of history, our understanding of the deep ocean was limited to samples dredged from the seafloor or occasional, brief descents in rudimentary diving bells. The immense pressure, pervasive darkness, and icy cold presented insurmountable barriers. This began to change significantly in the mid-20th century with the dawn of deep-diving submersibles. One of the earliest and most iconic manned submersibles was the bathyscaphe Trieste. In 1960, piloted by Jacques Piccard and Don Walsh, the Trieste achieved the incredible feat of reaching the deepest known point on Earth, the Challenger Deep in the Mariana Trench, descending to a record-breaking 10,916 meters. This monumental dive proved that humans could physically withstand the immense pressures of the Hadal Zone, albeit within the confines of a heavily reinforced spherical pressure hull. Manned submersibles, such as the famous DSV Alvin operated by Woods Hole Oceanographic Institution, or Japan's Shinkai 6500, allow human scientists to directly observe, collect samples, and operate equipment on the seafloor, providing invaluable real-time insights and the ability to react to unexpected discoveries. The Alvin, for example, was instrumental in the discovery of hydrothermal vents in 1977. More recently, private initiatives, like James Cameron's solo dive in the Deep-Sea Challenger to the Challenger Deep in 2012, continue to push the boundaries of manned exploration. While manned submersibles offer the unique advantage of direct human presence and adaptability, they are costly to operate, have limited dive times, and carry inherent risks. To overcome some of these limitations, marine engineers developed remotely operated vehicles, or ROVs. These are uncrewed underwater robots tethered to a surface support ship by a long cable, known as an umbilical. This umbilical provides power, transmits data video, sonar, sensor readings back to the ship in real time, and allows operators on the surface to control the ROV's movements and manipulate its instruments. The advantages of ROVs are significant. They can operate for much longer durations than manned submersibles, limited only by the ship's endurance. They remove human risk from the extreme environment. Equipped with high-definition cameras, powerful lights, robotic manipulator arms, and an array of scientific sensors for temperature, chemistry, current, etc., ROVs have become the workhorses of deep-sea exploration. They allow scientists to explore vast areas, collect precise samples of water, sediment, and organisms, deploy experiments, and conduct detailed surveys of the seafloor. Examples include the ROV Jason Woods Hole Oceanographic Institution, ROV Hercules Ocean Exploration Trust's Nautilus program, and ROV Tiburon Monterey Bay Aquarium Research Institute, MBARI. ROVs have been instrumental in discovering new species, investigating hydrothermal vents, and even exploring shipwrecks like the Titanic. Their real-time visual feedback makes them incredibly engaging tools for both scientific research and public outreach. The latest frontier in deep-sea technology is the Autonomous Underwater Vehicle, or AUV. Unlike ROVs, AUVs are untethered. They are pre-programmed with a mission plan and navigate independently, collecting data using onboard sensors. They can cover vast distances and operate for extended periods, from days to months, without human intervention. Once their mission is complete, they return to a predetermined location to upload their data and be recovered. AUVs are particularly valuable for mapping large areas of the seafloor, conducting systematic surveys, and exploring regions that are difficult or dangerous for manned vehicles or ROVs. They are quieter than ROVs, which can be advantageous for studying marine life sensitive to noise. While they lack the real-time human control and precise manipulation capabilities of an ROV, their ability to conduct broad, efficient surveys makes them indispensable for understanding the vast, unexplored expanses of the deep. Examples include MBARI's long-range AUVs for mapping methane seeps, or the Sentry AUV, which played a crucial role in locating the hydrothermal vents of the Lost City. The development of these technologies has not only expanded our reach, but also our sensory perception of the deep. Advanced sonar systems on these vehicles can map the seafloor in incredible detail, revealing hidden mountains, valleys, and fault lines. Specialized chemical sensors can detect plumes from hydrothermal vents or cold seeps, guiding explorers to new discoveries. High-resolution cameras, combined with powerful lighting, bring the alien landscapes and bizarre creatures of the abyss into vivid focus. These technological platforms are often used in combination during deep-sea expeditions. AUVs might conduct broad-scale mapping to identify areas of interest. Then, an ROV can be deployed to conduct detailed visual surveys and precise sampling in those specific areas. Finally, a manned submersible might be used for direct human observation and interaction with a particularly significant discovery. The ongoing evolution of deep-sea exploration technology continues to push the boundaries of what is possible. Next-generation vehicles are being designed to dive deeper, stay longer, carry more sophisticated sensors, and operate with even greater autonomy. These advancements promise to unlock even more of the deep ocean's uncharted mysteries, bringing us closer to a full understanding of Earth's largest and least known ecosystem. The technology isn't just a tool. It's our eyes, hands, and minds in a world that would otherwise remain forever hidden. Despite the incredible advancements in deep-sea technology that we just explored, it's a humbling fact that our understanding of the deep ocean remains incredibly limited. We have explored less than 5% of the global ocean, and a far smaller percentage of the deep sea has been observed in any detail. Vast stretches of the abyssal plains, countless seamounts, unexplored trenches, and even entire ocean basins remain largely unvisited by human eyes or robotic probes. This immense expanse represents the true frontier of exploration on Earth, holding an unimaginable wealth of future discoveries yet to come. Consider the sheer scale of the unexplored. The abyssal plains alone cover over half of Earth's surface, forming the largest continuous habitat on our planet. Yet, our sampling density in these areas is akin to taking a few handfuls of soil from random spots across an entire continent and claiming to understand its biodiversity. Each time a deep-sea expedition is mounted to a previously unvisited region, the likelihood of discovering new species is astonishingly high, often reaching over 50 for certain groups of invertebrates. This suggests that the true biodiversity of the deep ocean is vastly underestimated, potentially harboring millions of undiscovered species. many with unique adaptations and biochemical properties. What kinds of discoveries can we anticipate? Firstly, new species and novel biological adaptations. As we push into deeper, colder, or more chemically extreme environments, we expect to find organisms with increasingly specialized adaptations to pressure, temperature, and food scarcity. This might include new forms of bioluminescence, unprecedented metabolic pathways, or entirely new modes of life, yet unknown to science. The discovery of Ostex bone-eating worms at Whale Falls just 2 decades ago demonstrates how recently fundamental new animal types are being found. The deeper we go into the Hadal Zone, the more we learn about the physiological limits of life, and each new species found there pushes those boundaries further. Secondly, the discovery of new chemosynthetic ecosystems. While hydrothermal vents and cold seeps are now well known, scientists continue to find variations and entirely new types of these chemosynthetic oases. For instance, cold seeps powered by methane derived from gas hydrate dissociation, or seeps rich in hydrogen sulfide from processes other than deep burial of organic matter. There's also growing evidence of subseafloor communities microbes living within the oceanic crust itself, fueled by geochemical reactions, a truly deep biosphere that is largely unexamined but could represent a significant portion of Earth's biomass. As exploration technology improves, we may discover more lost cities, unique, low-temperature hydrothermal vent systems that operate on different chemical principles. Thirdly, understanding deep-sea geological processes. The deep ocean floor is a dynamic landscape, shaped by plate tectonics, volcanism, and seismic activity. High-resolution mapping with AUVs is constantly revealing new seamounts, submarine canyons, active fault lines, and vast abyssal plains dotted with manganese nodules. Many of these features influence deep-sea currents and act as hotspots for biodiversity. Investigating these geological features can provide crucial insights into Earth's internal processes, volcanic activity, and the formation of mineral resources. Fourthly, connections to climate change and global cycles. The deep ocean plays a critical, yet poorly understood, role in regulating Earth's climate. It is the largest active carbon sink, absorbing vast amounts of atmospheric carbon dioxide. How deep-sea ecosystems respond to ocean warming, acidification, and deoxygenation is a major area of future research. We need to understand how changes in surface productivity will impact the deep-sea food web, and how the stability of methane hydrates at cold seeps might be affected by warming waters, potentially leading to massive methane releases. Future exploration will be vital for monitoring these changes and developing accurate climate models. Finally, the deep ocean holds immense potential for biotechnology and medicine. The extremophiles living in vents and seeps produce unique enzymes and biochemical compounds adapted to extreme conditions. These extremozymes are of great interest for industrial processes like detergents that work in cold water or high-temperature industrial reactions, and for drug discovery. The potential for novel antibiotics, anti-cancer agents, or other pharmaceuticals from deep-sea organisms is largely untapped. Every new species found represents a new genetic library, a new set of biochemical possibilities. To achieve these discoveries, future exploration will rely on increasingly sophisticated technologies. We can anticipate deeper diving AUVs and ROVs, capable of reaching the very deepest parts of the trenches more frequently and efficiently. Longer duration missions, untethered vehicles that can spend months or even years exploring vast areas autonomously, transmitting data periodically. Artificial intelligence and machine learning. To process the enormous datasets collected by these vehicles, identify patterns, and even guide autonomous exploration. Miniaturization and swarms. Fleets of small, highly mobile, and interconnected robots that can collectively explore large areas or specific complex features. Advanced sensing. New sensors capable of detecting chemical signatures, genetic material, and biological interactions in ways we can only dream of today. In situ observatories. Permanent or long-term deep-sea observatories that continuously monitor environmental parameters and biological activity, providing a temporal dimension to our understanding. The deep ocean is more than just a place. It's a profound reservoir of knowledge, a living testament to the resilience of life, and a critical component of our planet's operating system. The discoveries yet to come in these uncharted territories will not only satisfy our innate curiosity, but are essential for understanding the future of life on Earth. The lure of the abyss is not just about what we have found, but what remains to be found. A vast, silent, and ancient world waiting patiently to reveal its secrets. We have journeyed through the incredible depths of the deep ocean, marveling at its unique ecosystems, astonishing adaptations, and the vast, uncharted territories that still await discovery. However, it is crucial to recognize that even this remote, seemingly untouched realm is not immune to the pervasive influence of human activity. The deep ocean faces a growing number of threats, and safeguarding its fragile, slow-recovering ecosystems is becoming an urgent conservation imperative. Understanding these impacts and the need for protective measures is vital to preserve this last true frontier. One of the most immediate and visible threats to the deep ocean is pollution, particularly plastic pollution. While often associated with surface waters, plastics sink. Microplastics and even larger plastic debris have been found in the deepest trenches on Earth, including the Mariana Trench. These plastics can be ingested by deep-sea organisms, leading to internal injuries, starvation, or exposure to toxic chemicals that leach from the plastic. The slow degradation rate of plastics in the cold, dark deep means that once introduced, they can persist for centuries, accumulating and causing long-term damage. Beyond plastics, other forms of pollution, such as chemical runoff and discarded fishing gear ghost gear, also pose significant threats, entangling marine life and polluting habitats. Another escalating threat is deep-sea mining. As easily accessible terrestrial mineral deposits dwindle, attention is turning to the vast mineral resources found on the deep seafloor. These include polymetallic nodules on abyssal plains, rich in nickel, copper, manganese, and cobalt. Polymetallic sulfides at hydrothermal vents, containing copper, zinc, gold and silver, and ferromanganese crusts on seamounts, rich in cobalt and rare earth elements. Several companies and nations are actively pursuing permits and developing technologies for deep-sea mining. The potential impacts of deep-sea mining are severe and long-lasting. The process itself involves specialized vehicles collecting or scraping minerals from the seafloor, generating plumes of sediment that can spread for vast distances, smothering sessile organisms, reducing water clarity, and disrupting feeding patterns. The removal of geological features like hydrothermal vents or seamount crusts would destroy unique habitats that have taken millennia to form. Given the incredibly slow growth rates and long lifespans of deep-sea organisms, and the isolation of many deep-sea communities, recovery from mining disturbances could take hundreds, if not thousands, of years, if it occurs at all. The loss of these unique ecosystems and potentially undiscovered species would be irreversible. Deep-sea fishing also poses a significant threat. While target species are often shallower, bycatch from trawling and longlining can damage deep-sea corals, sponges, and other fragile benthic habitats. Bottom trawling, in particular, involves dragging heavy nets and weights across the seafloor, causing extensive and long-term damage to slow-growing, sensitive ecosystems that are highly vulnerable to physical disturbance. Many deep-sea fish are slow-growing and late to reproduce, making their populations highly susceptible to overfishing and slow to recover. Climate change impacts are also reaching the deep ocean, although sometimes in less direct ways than at the surface. Ocean warming. While the deep sea is largely insulated from rapid surface warming, changes in deep water formation in polar regions can subtly alter deep-sea temperatures over long timescales, potentially affecting cold-adapted species. Ocean acidification. The ocean absorbs a significant portion of atmospheric carbon dioxide, leading to ocean acidification. While most pronounced in surface waters, this acidification also affects the deep sea, particularly in colder, high-pressure environments where calcium carbonate essential for shells and skeletons dissolves more readily. This can impact deep-sea corals, mollusks, and other calcifying organisms. Deoxygenation. Warming surface waters and changes in ocean circulation can lead to reduced oxygen levels in the deep ocean, expanding oxygen minimum zones. Many deep-sea creatures are highly sensitive to oxygen levels, and deoxygenation can create dead zones, limiting habitat and stressing organisms. Changes in food supply. Climate change impacts on surface productivity, e.g., changes in phytoplankton blooms will directly affect the amount of marine snow reaching the deep sea, potentially altering food availability for entire abyssal ecosystems. Bioprospecting. The search for novel genes, molecules, and compounds from deep-sea organisms for industrial, pharmaceutical, or other commercial uses also presents a potential threat if not conducted responsibly. While it holds immense promise for new discoveries, unregulated collection could deplete populations of rare species or damage sensitive habitats. Given these mounting threats, safeguarding the abyss has become a critical focus for marine conservation. This involves several key strategies: establishing marine protected areas (MPAs) in the deep sea, designating specific areas as protected zones where extractive activities like fishing or mining are prohibited is a crucial step. This requires international cooperation, as many deep-sea areas lie beyond national jurisdictions in the high seas. Developing robust international regulations. Organizations like the International Seabed Authority, ISA, are tasked with regulating deep-sea mining in areas beyond national jurisdiction. Developing and enforcing strict environmental regulations, including baseline environmental assessments and monitoring, is essential to minimize impacts. Promoting sustainable fisheries practices. Implementing stricter quotas, regulating deep-sea trawling, and using less destructive fishing methods can help protect vulnerable deep-sea populations and habitats. Reducing pollution at its source. Addressing plastic pollution, chemical runoff, and other contaminants at their terrestrial and coastal origins is vital to prevent their entry into the deep ocean. Investing in research and monitoring. We cannot protect what we do not understand. Continued investment in deep-sea exploration, scientific research, and long-term monitoring programs is essential to establish baselines, track changes, and inform conservation strategies. Raising public awareness. Educating the public about the vital importance and vulnerability of the deep ocean is crucial to foster support for its conservation. The out of sight, out of mind mentality must be overcome. The deep ocean is not an infinite resource, nor an impervious void. It is a fragile, ancient, and highly interconnected ecosystem that plays a fundamental role in global planetary processes. As our technological capabilities allow us to reach deeper, we bear an increasing responsibility to understand, value, and protect this extraordinary realm before human activities inflict irreversible damage on its uncharted mysteries. The future of the deep abyss, and ultimately our planet's health, depends on our collective stewardship. As we bring our comprehensive journey into the deep ocean and its uncharted mysteries to a close, it's fitting to reflect on the profound allure and enduring enigma of this extraordinary realm. We began by acknowledging its vastness and our limited understanding, and through our exploration, we've only deepened our appreciation for its complexity, its beauty, and its fundamental importance to our planet. We plunged through the sunlit zone, where photosynthesis fuels abundant life, and descended into the dimming light of the twilight zone, the mesopelagic, where the first whispers of bioluminescence and grand daily migrations hint at a hidden world. Further down, into the true midnight zone of the bathypelagic and the expansive plains of the abyssal pelagic, we encountered life adapted to crushing pressures, icy cold, and perpetual darkness, surviving on the sparse rain of marine snow. And then, to the ultimate depths, the hadal zone, where life defies logic, thriving in trenches under forces that challenge the very limits of biology. We explored the ingenious ways life adapts to these extreme conditions. The incredible chemistry of bioluminescence, serving as a language of light for communication, hunting, and defense. The physiological marvels of slow metabolisms, gelatinous bodies, and pressure-stabilizing molecules. And the fascinating phenomenon of deep-sea gigantism and dwarfism, showcasing the diverse evolutionary paths to survival. Our journey then took us to the truly revolutionary discoveries of deep-sea ecosystems powered by Earth's internal energy. Hydrothermal vents, cold seeps, and even the transient oases of Whale Falls revealed that life can flourish entirely independently of sunlight, drawing energy from chemical reactions. These chemosynthetic communities have not only rewritten our understanding of Earth's biology, but have also offered tantalizing clues about the origin of life and the potential for life on other ocean worlds in our solar system. We acknowledged that our ability to unravel these mysteries is a testament to human ingenuity. With advanced technologies like manned submersibles, remotely operated vehicles, and autonomous underwater vehicles acting as our eyes, hands, and scientific instruments in this alien environment. Yet, for all our technological prowess, we've only scratched the surface. The vast majority of the deep ocean remains unexplored, holding untold numbers of undiscovered species, unknown ecosystems, and profound scientific insights that will continue to challenge and expand our knowledge. But perhaps the most critical takeaway from our exploration is the urgent need for stewardship. This magnificent, ancient, and fragile realm is not immune to human impacts. Plastic pollution, the looming threat of deep-sea mining, the lingering effects of historical fishing practices, and the pervasive, long-term consequences of climate change, ocean warming, acidification, and deoxygenation are all reaching into the abyss. The slow pace of life in the deep means that these ecosystems are incredibly vulnerable, and recovery, if at all possible, would take centuries or millennia. The deep ocean is more than just a distant, exotic place. It is an integral and vital component of our living planet. It regulates our climate, cycles essential nutrients, and harbors an unimaginable reservoir of biodiversity and potential biotechnological breakthroughs. Its health is inextricably linked to the health of the entire global ocean, and by extension, to our own well-being. The enduring mystery of the deep is its greatest strength and its greatest vulnerability. Its remoteness has protected it for eons, but now, as our reach extends, so does our responsibility. The deep ocean compels us to ponder not just what is hidden beneath the waves, but how we, as a species, will choose to interact with this final frontier. Will we explore it with reverence and protect it with diligence? Or will we exploit its resources before we even fully understand its intricate workings? Let the silence and vastness of the deep ocean not be a barrier to our concern, but rather an invitation to a profound sense of wonder and respect. Let the bizarre beauty of its creatures inspire our imagination and fuel our desire to learn more. And let the uncharted mysteries serve as a powerful call to action, urging us to safeguard this invaluable global heritage for generations to come. The deep ocean is still largely unknown, but it is undeniably important. Its secrets are waiting patiently for us to understand.